Subscribe to Simplified Biology channel and press the bell icon to get notified when a new video is uploaded. Hello friends, welcome to Simplified Biology. Today's topic is Mendelian Inheritance. A quick revision for NEET. Mendelian Inheritance. Now, Sir Gregor Johann Mendel was an Austrian monk who performed his experiments in the kitchen garden of the monastery in which he lived. He started his experiments in the year 1856, published them in 1865, and died in the year 1884. His work was rediscovered by three scientists separately in the year 1900, and they were Hugo de Veris, Correns, and Cheshire. After which, Mendel came to be known as father of genetics. Now, there are some terms that should be clear concept-wise. Allele. Allele means two or more alternatives of a gene, or two or more forms of a gene. Pure or true. Pure or true breeding is actually the other word for homozygous. But they can be best explained as pure breeding or true breeding organism is the one which upon self-fertilization produces offspring completely identical to itself. And this is only possible if it is in homozygous condition. Hybrid, also known as heterozygous or the other word for heterozygous. Hybrid is an organism which has at least one gene in heterozygous condition. Reciprocal cross. Reciprocal cross of any cross is the characters taken are the same, but they are taken in different sex. Like if a tall female is crossed with a dwarf male, its reciprocal cross will be tall male and dwarf female. Test cross. Test cross is crossing the offspring with an homozygous recessive organism. Homozygous recessive organism. This is done to find the genotype of the offspring. Now, Mendel used garden pea. Garden pea, that is Pisum sativum, as the plant for his experiments. And he used 7 pairs of contrasting characters. Now, to remember these characters, the best way is, first is about the plant, that is the height of the plant, whether it is tall or dwarf. Next comes to the reproductive part of the plant, that is the flower, color of the flower, and also its position. Flower develops into fruit. So next comes the fruit, which in pea is referred as pod, that is shape of the pod and color of the pod. And developing inside the fruit is the seed. So next is the seed, shape of the seed and the color of the seed. So these are the 7 characters that Mendel took into consideration. The monohybrid cross done by Mendel was by taking the height of the plant. He crossed a pure tall with a pure dwarf. In the F1 generation, he found that all were tall. When the F1 tall is self-fertilized, he got the ratio 3:1. That is 3 tall and 1 dwarf in the F2 generation. Now, after coming across the dwarf character in the F2 generation, Mendel concluded that in the F1 generation, along with the tall, the dwarf is also present. And of the two, only one expresses, the one which expresses is dominant and the other one is recessive. This came to be known as the law of dominance. He then self-fertilized the F2 generation. When he self-fertilized the F2 generation, he saw that of the 3 tall, 1 tall upon self-fertilization gave all tall offsprings. An organism upon self-fertilization, when produces offspring completely identical to themselves, then they are referred as pure. So this 1 tall is pure. The other 2 tall again gave the ratio 3:1, which means this tall has dwarf in it. That is, it is hybrid. And the 1 dwarf produce all dwarf offsprings, which means this too is pure. Now this showed that the 3:1 ratio is actually 1:2:1. 3:1 is the phenotypic ratio, while 1:2:1 is the genotypic ratio. After which Mendel gave his law of segregation, also known as purity of gametes. Now this law states that this is pure tall is crossed with pure dwarf. In the F1 generation, all are tall. Now this law states that in a heterozygous organism, the dominant and the recessive gene will stay together throughout the life without mixing with each other. And during gamete formation, they separated from each other and entered the separate gametes. So by self-fertilizing the F1 generation, he showed the phenotypic and the genotypic ratio. This is pure tall, hybrid tall, and pure dwarf. Now he then performed the dihybrid cross by taking the color and shape of seeds. He crossed yellow round seeds with green and wrinkled seeds. In the F1 generation, he found that all the seeds were yellow and round. When the F1 generation is self-fertilized, he got the ratio 9:3:3:1. 9 of which were yellow and round, means both dominant. 3 were yellow and wrinkled, means 1 is dominant and 1 is recessive. 3 are green and round, 1 is dominant and 1 is recessive, and 1 is green and wrinkled. After which Mendel gave the law of independent assortment. Now this law states that in a heterozygous organism, when 2 or more characters are taken into consideration during gamete formation, members of each pair will separate from each other and they will form all possible combinations in the gamete. So to find out the number of gametes produced, the formula is 2 to the power n, where n represents number of characters in heterozygous condition. Remember, it is heterozygous condition. So in this case, the number of gametes will be 2 to the power 3, as there are 3 characters in heterozygous condition. That is, 8 types of gametes are produced. Now the dihybrid phenotypic ratio is always 9:3:3:1. In 9 cases, both the genes, one dominant of both the genes are present. In 3 cases, one is dominant and the other one is recessive. In the other 3 cases, the first becomes recessive and the other is dominant, and in one, both are recessive. While the dihybrid genotypic ratio is 1:1:2:2:4:2:2:1:1. Means there are 4 ones, 4 twos and 1 4. Now wherever the case is 1, both the genes are homozygous. It is this condition, this, and this. And where the case is 2, one gene is in homozygous condition and the other one is heterozygous condition. This way, this is homozygous, this is heterozygous. Now in this case, the first is heterozygous and the second one is homozygous. And wherever the condition is 4, both are heterozygous. Remember, the genotypic ratio never changes. The points to remember to solve MCQs on Mendel crosses are: the monohybrid cross phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Dihybrid cross, the phenotypic ratio is 3:1 into 3:1, that is 9:3:3:1, and the genotypic ratio is 1:2:1 into 1:2:1. Trihybrid cross, the phenotypic ratio becomes 9:3:3:1 into 1:3:1. Now always remember, whatever the conditions may be, the genotypic ratio never changes. And this helps us in solving a lot of questions. Now while solving questions, these things should be clear in your mind, that when a homozygous tall is crossed with a homozygous dwarf, a heterozygous tall is produced. When a heterozygous tall is crossed with a homozygous tall, that is a test cross like pattern, it is always 1:1. When the tall is homozygous and the other one is heterozygous, in this case too, the phenotypic ratio, phenotypic all will be tall, but the genotypic will be 1:1. In this case also it is 1:1, and in this case also it is 1:1. And when both are heterozygous, the ratio is 1:2:1. Now, how the genotypic ratio of dihybrid cross and the genotypic ratio of a monohybrid cross can be used to solve a question like this. Find the F2 ratio of the following offsprings. Both homozygous dominant A, heterozygous B, homozygous recessive A and heterozygous B, heterozygous A, heterozygous B, homozygous recessive A and B. Now remember the 1:1:2:2:4:2:1:1 ratio. Now in that case, where both the homozygous, the condition is 1. One homozygous and one heterozygous, the condition is 2. Again, one homozygous, one heterozygous, the condition is 2. Both heterozygous, the condition is 4. And both homozygous, the condition is 1. So, the ratio of these offsprings are 1:2:2:4:1. Now, these can also be solved by using the monohybrid genotypic ratio. That is by separating these. Homozygous, the genotypic ratio, monohybrid is 1. So 1:1. Homozygous 1 and heterozygous is 2. Homozygous 1, heterozygous 2, heterozygous 2, heterozygous 2, homozygous 1, homozygous 1. Means the ratio is 1:2:2:4:1. Now if you are given a dihybrid cross where one gene follows the law of dominance and the other gene is codominance or codominant, then to find the ratio, phenotypic ratio, it will be 3:1 into 1:2:1. As in codominance, the phenotypic and the genotypic ratio are the same. That is 1:2:1. Now let's see another question. Now in a cross where homozygous dominant A and heterozygous B, a dihybrid cross, is crossed with the homozygous recessive A and heterozygous B. You have to find the ratio of the following offsprings. Best way is you have to remember that we have to cross these two separately, or you have to remember this. In this case, always the condition will be this, while in this case we get the ratio 1:2:1. And when you multiply them with this, it becomes 1, 2, and 1. Means this is 1, this is 2, this is 1, and this is 0. The ratio of the offspring is 1:2:1:0. Now another question. Which one of the following crosses show phenotypic ratio 3:1? Now remember, phenotypic ratio 3:1 is a monohybrid phenotypic ratio. So let's go to the options. In the first option, we see that both are heterozygous, or it is a dihybrid cross where the phenotypic ratio is 9:3:3:1. In the second option, we'll find this condition is seen. A is heterozygous crossed with homozygous A, means genotypically they may be different, but phenotypically they are the same. While when homozygous B is crossed with heterozygous B, dominant B, in this case, two types of offsprings will be seen, one dominant and one recessive. So these both together will not give the ratio 3:1. In the third option, heterozygous A crossed with heterozygous A, we get the ratio 3:1, while in case when B is this, they are all will be the homozygous recessive B. In this case, we see the ratio 3:1. While in the fourth option, first is a heterozygous AB is crossed with a homozygous AB. It's just like a dihybrid cross, where 4 types of offsprings will be produced in the ratio 1:1:1:1:1. So the correct option in this case is C. Last question, a slightly different one. A man with A blood group and Rh positive marries a woman with A and Rh negative blood group. They have a son of O negative blood group. Now this is a type of trihybrid cross. Father is A, means father has X and Y chromosome and is positive. Mother is A and is negative, and the son is O and negative. Now for O condition, the recessive I gene is present in homozygous condition, and for the negative blood group, the recessive gene is present in homozygous condition. Now this shows that both the father and the mother have the A gene in heterozygous condition. Only then it is possible for a child to have a O blood group. And since the child is negative, having the recessive gene in homozygous condition, which means the father is heterozygous for the positive gene and mother has the homozygous recessive gene. The question is, what is the probability that the next child will be male and O negative? Up till now we have seen only 2 genes. To find out whether the next child will be male and O negative, we have to take them as father has an X and Y chromosome while mother has XX chromosome. So when XY is crossed with XX, means there are 50% chances to have a male child. Means in half cases a male child is formed. And when both parents are heterozygous A, means in 1/4 cases there are chances of having O blood group. And in case of Rh positive and Rh negative, father is heterozygous for the Rh positive gene and mother has the homozygous recessive gene, and the child to have the homozygous recessive gene, half the chances is there. So for the next child to be male and O negative, the chances are 1/2 into 1/4 into 1/2. That is 1/16. So the answer here is 1/16. That's all for today. Please do like, subscribe and comment, and also visit our website by clicking on the link given below. Thank you for watching.